Thank you all for joining us for the first event of this semester's Basic Science Lights the Way series. I think it's particularly appropriate because I'm sure you all have noticed that this is Nobel Week, and that's a big week around Berkeley. And we are pretty happy because both of our divisions have been implicated in the prizes that were awarded yesterday and today. Maybe you noticed that Gary Ruvkin, who won the Physiology or Medicine Nobel Prize yesterday for the discovery of microRNA, he was a biophysics major back in 1973. And we just posted something in the chat about him. And then yesterday, or today, John Hopfield, who won the prize along with Jeffrey Hinton for artificial intelligence. He was not an alum, but he spent his first faculty years in the physics department back in the 60s. So I like to talk about these prizes because they're both fantastic examples of basic curiosity-driven science, precisely what we're celebrating with this series and what we've been celebrating for several years now in this series. So with that, let's turn to tonight. We are joined by some of our superstar faculty members and students from both divisions of biological sciences and mathematical and physical sciences. From a physicist who alters the fundamental property of materials to a neuroscientist who activates synapses in the brain, and to an astronomer who unveils new stars and galaxies. The thing that they have in common this evening is that each of them use light as a primary tool for discovery. Light serves as a cornerstone of basic science research, illuminating our understanding of the natural world in profound ways. As an electromagnetic wave, light possesses unique properties that allow it to interact with matter, making it an essential tool for exploration across various scientific disciplines. By enabling us to observe, analyze, and manipulate phenomena at both macroscopic and microscopic scales, light not only enhances our knowledge of fundamental concepts, but also paves the way for groundbreaking advancements that shape our future. To help us explore this topic further, I would like to introduce our moderator, Jessica Liu, who's a professor of astronomy, and she is currently chair of the Astronomy Department. That's a very big job. Jessica's also been one of our presenters at a past basic science event. Jessica runs the Moving Universe Lab, which searches for free-floating stellar mass black holes in our Milky Way galaxy using gravitational lensing. These areas of research require sharp images from both space telescopes and ground-based telescopes equipped with adaptive optics to correct for the blurring of the Earth's atmosphere. She's currently exploring new technologies for manipulating light and improving telescopes to deliver the sharpest ever visible images of the universe. So you can imagine why she's a perfect moderator tonight. Thanks for being here, Jessica. Take it away.
Thank you so much. And can you hear me okay?
Yep.
Excellent. Well, thank you, Maria. I'm happy to be moderating tonight's event, exploring the role of light in scientific discovery. I use light in many ways to learn about the universe, and I'll start by reminding you what light is. Let me get the slides going here. Light is an electromagnetic wave that can come with a variety of wavelengths or colors. You're familiar with optical light, which has a wavelength comparable to the size of bacteria. Radio and infrared waves are longer wavelengths, while X-rays have short wavelengths the size of a single atom. Light can be manipulated. We can use lenses and mirrors to fold and focus light. Here's an example in this slide of the Keck telescopes. This is a 10-meter telescope in size, and it focuses light from distant objects onto cameras that are the size of your cell phone. So it's critical to be able to manipulate light in this way to see those distant galaxies. Unfortunately, light waves can be distorted. Our largest telescopes on Earth deliver blurry images as a result of the Earth's atmosphere. So I like the Earth's atmosphere. It provides us a lovely cocoon for being able to breathe, but it also makes our stars twinkle and become blurry. We help build something called adaptive optics to take the twinkle out of the stars. And adaptive optics works by sensing distortions in the incoming wavefront, as shown here, and correcting those distortions 1,000 times a second with a deformable mirror shown in the middle. And then on the right is the corrected wavefront. So you can take those blurry, twinkly images of the stars that you see in the lower left corner and turn them into beautiful, sharp images, limited only by the diffraction of our size of our telescope. And finally, objects in the universe can also manipulate light. Oh, sorry, let me rephrase, step back for just a minute. I forgot. Light can also be used to excite or change matter. And this is going to be critical for tonight's speakers. So we can use light to change the state of matter. For instance, in this case, we're using a laser. You see the laser beacons coming out from the top of the telescope. This laser is a pure coherent beam of light at a single wavelength, and we're using it to excite molecules. That can be in the brain, or in this case, in the atmosphere. And this shows this laser that's tuned to excite sodium atoms about 90 kilometers above the ground. Those sodium atoms then glow, and they look like a star. And we can use this artificial star generated by light to drive our adaptive optics system and explore the universe with sharp images. And so then finally, objects in the universe can also manipulate light. We can't forget that. And we can see some of those manipulations. In this animation, I'm showing you an example of a black hole, that's the black dot on the left, acting as a lens itself. Its own gravity lenses a background star as it passes by. That's shown in orange. So what's on left is the schematic. What's on the right is a simulation of how we might be able to observe this phenomena of the object in the universe manipulating light. It's not something we can see today, but we're working to improve our adaptive optic systems on large telescopes to see this effect in the future. And you'll hear more about that from one of our speakers. So this evening, we have 3 speakers working in vastly different areas, but they all utilize light in their research, manipulating and imaging in 3 dimensions, using light to tomographically map 3-dimensional structures. And I think you'll be very excited by all of their talks. So thank you for joining us today. Once we get underway, we encourage you all to post your questions to our speakers in the chat box. And at the end of each talk, we'll try to address a few of the questions, and we have lots of time at the end for more questions. So I'd like to turn to our first speaker. Our first speaker this evening is Max Huber, a graduate student in the Department of Physics. He works in the lab of physics professor Alessandro Lazzara. The Lazzara lab interests are in exploring the science and technology of sustainable materials, quantum materials, and novel biological processes. Through development and application of advanced scientific tools, coherent optical laser drives, and artificial intelligence, her lab is at the forefront of material science and condensed matter research. Max uses time-resolved, angle-resolved photoemission spectroscopy to study photo-induced phase transitions. And you'll hear more of that in his talk. So Max, thank you for joining us this evening. And can you tell us a bit about your research and projects you're working on?
Yes, hello everyone. Thank you for the nice introduction. And thank you for giving me the chance to talk about some research we're doing in my group, and in particular, how we can use light to control materials properties. So one of the biggest problems that we have in society is that we generate huge amounts of waste. And even though we're running out of a lot of valuable materials like lithium and other rare earth metals, only a fraction of it is recycled. So there's a huge demand that in newer technologies, we become more resourceful and more sustainable in what we do. So to explore this more, let's go a step back first and look into what are the kind of materials that we're actually using. And a rough way to characterize different solids is by the ability to conduct electricity. So on the one extreme, we have metals, which are excellent conductors. And on the other hand, we have insulators, which conduct electricity very poorly. If one looks at this from a very fundamental point of view, then there's clear differences in the electronic structure. So in every solid, one can kind of define the highest electronic states that are occupied by electrons. We call this a valence band. And the lowest lying states that are not occupied by electrons, we call this a conduction band. And now in a metal, these 2 bands overlap, and it's very easy for the electrons that are in a valence band to hop up into the conduction band and move around freely and conduct the electricity. Versus in insulators, there's a pretty large gap between the two bands. So these electrons are basically locked in the valence band, they cannot move and they cannot carry any electricity. And so nowadays technology, we have many different materials with many different properties, and we can engineer this kind of gap and conductivity very precisely. But a big problem is that once we've kind of made this material, the properties are static and we cannot change it anymore. So it would be way more efficient for a lot of applications if we were able to kind of switch between system being metallic and insulating. So to kind of illustrate this, we can think about it as like a light switch where we can switch between being insulating and being metallic, depending on what we need right now. So the question is, how can we achieve such a goal? And like as in many situations, we can draw inspiration from nature. Photosynthesis is one of the most important processes on Earth. And here, like plants use solar light that comes from the sun to convert carbon dioxide into oxygen. And kind of, this is something similar what we want to do. We want to do light, or in particular laser pulses, to control our materials properties and switch between insulators and metals. Laser pulses are particularly nice because we can control them very well, and we can make them extremely short. So like in the data that I will show you later, and for the experiments in my lab, we use laser pulses that are 50 femtoseconds long. So 1 femtosecond is 10 to the minus 15 seconds. That's like a billionth of a millionth of a second. So it's like extremely short time scan that no electric pulse or anything else could do. So you could really excite extremely fast processes with a laser. So now let's get to some examples in particular. The first material that I want to show today is titanium diselenide. It's like a picture, it forms these nice little crystals that are very nice for us to measure. And with the technique that I'm using in the lab, we can now directly visualize the electronic structure of this material. So the spectrum then that we take looks something like this. So blue means that there's basically no intensity, and red means that there's high intensity. So that means we have pretty high intensity of states here below the Fermi level, but nothing at the Fermi level. And if we go back to our band model that I showed earlier, then we can see if there's no intensity of states at the Fermi level, that means that the system is a rather poor conductor and more on the insulating side. So now we can go ahead and take this material and excite it with a laser pulse. And if we do so and we wait 100 femtoseconds, then we can see that the electronic structure drastically changes, and now we have a lot of states that go through the Fermi level. So we really drive a transition from an insulating into a metallic state on an extremely fast time scale. Like, if you remember 100 femtoseconds, that's 110 to the minus 15 seconds. And another interesting thing is we can now wait a little bit longer. So if we go to a little bit later time to about 600 femtoseconds, then we can see that this gap is almost open again, and the states at the Fermi level decrease significantly. So in this kind of material, we can really drive extremely fast transition from a gapped insulating state to a metallic state and back to an insulating state. So this is very interesting for especially like high-speed computing in the future where we need very fast processing speeds. However, it's not always desirable that like a metallic state goes away that fast. Like sometimes we'd also like it to stay longer. And this actually can be realized in a different material called tantalum disulfide. I also attached a picture here. You can see it looks quite similar, but it's different compounds, slightly different structure. And again, we can go ahead and measure the electronic structure here. And similar to titanium diselenide, there's not a lot of states at the Fermi level. So it is insulating in the ground state and gapped. And we can go ahead again and excite it with a laser pulse. And it again becomes metallic. You can clearly see there's now a lot of intensity at the Fermi level. But the characteristic difference to the previous example is that this metallic state is really stable variety 1 millisecond to 1 day, but actually the state, if the temperature is low enough, can be stable for weeks or months if you want. So this is a really stable state that stays that way. But then if we increase the temperature, it goes away again and it reverts back into this gapped state. And so this is a amazing opportunity to realize a lot of interesting physics devices in the future. For example, if you think about transistors, which is like the basic building block of all computers that we use nowadays, they can regulate current by switching between conducting and insulating. But you always need to apply a voltage to do so. So if the electricity turns completely off, the system loses all memory, and then that leads to these kind of complicated computer architectures that we have today. So with a material like this, it would remember its state. Like once it's metallic, it stays metallic, and if it goes back to insulating, it stays insulating. So this would lead to computers being far more efficient in the future, faster, and they can also be a lot smaller than these kind of complicated transistor devices. Another thing that we can do, because the state is stable, is we can go ahead and zoom in on like a little region of our sample, and then we can make our laser pulse very, very small and shoot it at different spots on our sample. And if we do so, we can get a map like this, where the black regions are metallic regions where we shot the laser, and the gray region is the background that is still insulating. So with this technique, we're really able to write metallic patterns on our surface on like a really small length scale. And then all of these metallic regions can get easily erased again if we just heat it up a little bit. So there's a lot of potential to create a lot of metallic structures in various shapes, however it is needed. And so that already brings me to my summary. I hope that I was able to convince you that laser pulses is a very efficient tool to switch between insulating and metallic states in the sample. And ultimately, this kind of ability that we're able to switch between different states in the same material will be very important in the future, that we can make technology that is more efficient in the sense that we need less energy to use it, but also more efficient in the sense that we need less material to build it in the first place. And then we can be more resourceful with the materials that we have on this planet. And with that, I thank you for your attention, and I'm happy to answer questions.
Thank you, Max. It was really interesting. I think we might have a few questions for you, but while we're waiting for everyone to post their questions in the chat, go ahead and post those now. I was curious, can any material be switched like the examples that you showed, or do these materials have to have special properties?
Yeah, that is a good question. Like, I didn't have too much time in the talk to go about this, but if you take like a normal metal or like a normal semiconductor like silicon, there's only very weak interaction between like the different electrons, for example. So if you excite those with a laser, then there's not too much that will be happening. So usually, like the materials that I showed that belong to like a class is called correlated materials. So there's a lot of important interaction effects between the electrons going on. And this can be easily disrupted with a laser because there's a lot of energy and stirs up a lot of things. So you do need some correlated materials to actually see these kind of changes.
Great. And what kinds of lasers, how are the lasers that you use to change these materials different than our everyday laser pointer?
So they're different in the sense that the pulses are a lot shorter. So like if you have like an everyday laser pointer, then they often still pulse, but they have like a way longer pulse length. And then the other thing is that we also focus them more and use higher powers to really see those changes. But in principle, there's some things you could even do with a laser pointer.
Excellent. All right, unless we have any other last minute questions. Okay, I think we're going to move on to our next speaker of the evening. I'll welcome a professor in the departments of neuroscience and molecular and cell biology, as well as director of Berkeley's Helen Wills Neuroscience Institute, Ehud Isacoff. So Professor Isacoff works on synapses, which are the connections between neurons, plasticity, the changes in connection strength at synapses, which mediate memory. He uses light both to read and write information to the brain. He discovered how the ion channels that generate the action potential sense membrane voltage, and he's now studying how receptors at synapses sense neurotransmitters. He co-invented the field of chemical optogenetics, which uses light to manipulate protein structures in order to control these neural signals. And he applies this research to neuronal diseases, including retinal diseases, to restore neural function. So Professor Isacoff, please tell us about your research.
Thanks, Jessica. Let me start with what it is that we'd like to understand. We'd like to understand the function of the human brain. And as you can see here, the human brain, which looks very familiar, is quite big. And if you've been reading the newspapers recently, then you may have seen that the first step to try and understand how the human brain works as a computer has been made recently in the complete connectome, as it's called, of the brain of the Drosophila fruit fly, which comparatively, I don't know if you can see this tiny little dot here, but that's the size of a Drosophila brain. So the Drosophila brain has 50,000 neurons and about 1,000 times as many connections between the neurons. The human brain, 100 billion neurons with 100 trillion connections. But the hope is that if you can understand the circuit diagram of these brains, then you'd be able to learn a lot about how they operate. And here's just an example of what the circuit diagram looks like for the fly brain. This is every neuron you can see here. And then if you look at individual neurons that are involved in, let's say, vision or movement of the fly, then what you do is you end up seeing subsets of cells, and you can see every connection between every cell. So you think that we're actually well on the way to understanding the fly brain from this, but there's a problem, and that is the physical connections that we can see are not reflective, when you look at who's connected to whom, of the strength of those connections, or how those connections change strength with experience. And so as a result, we're still stymied, even understanding the little fruit fly, in how it learns and remembers and thinks, which is what we want to understand about our own much, much larger brain. So what we would like to do in our lab is shed light, forgive the pun, on that problem. And we do this in two kinds of ways that Jessica alluded to earlier, both using light. One is to read the activity of the brain, and the other is to write activity to the brain. So here's an example of how this is done in our lab in fruit fly. You can, when you look at the larva, the maggot of the fruit fly, you can actually monitor its behavior in many, many animals at the same time. You can see how waves of activity go through individual neurons in the central nervous system. That's this picture here. Or in this movie here, you can see how waves of communication between one neuron and another happen going through the entire organism. And if you blow up something like this, you can look at individual parts of the connections and see how they turn on and off with time. And if you blow it up even more, then you can see actually what is the fundamental neurotransmission event of the nervous system, which is the release of a packet of neurotransmitter by one neuron that activates receptors in the responding neuron. Each light flash represents what we call a quantum of neurotransmission. The elementary synaptic transmission event that involves about 1,000 molecules of neurotransmitter. And the question is, like, what controls this release process? If we could understand that, then we'd be able to maybe paint on the neural circuit that I showed you earlier how strong every connection is and how it changes with experience. And to do that, we have to look even deeper and at a smaller scale. Now this is individual molecules in the neuron that's releasing the neurotransmitter of the apparatus, the molecular apparatus that is controlling the neurotransmitter release process. So what we can do is across scales visualize the molecules, the transmission events, how these happen over entire cells, how they happen throughout a circuit and transmission synapses of a common kind, how that works because of control of activity throughout the nervous system of series of neurons and how that mediates behavior. So this is light being used in the fly, neurons and synapses, in order to read activity in the nervous system. We're not just constrained to doing that in the fly. We can do it on that intermediate brain, the mouse, whose brain is much more similar to ours, although still vastly smaller. Here's a mouse. The mouse is running on a ball. The ball is made of styrofoam, and it's sitting on a cushion of air. So it can rotate as the animal runs in place. If the animal's thirsty, it can do something and receive a reward of water. We can have the mouse under a microscope so we can visualize the activity of neurons in its cortex. And this is a part of the cortex that's involved in motor coordination. So the motor behavior that you're seeing there is driven by the neural activity that you see here. And this is done with a kind of microscope that is able to slice optically. It's not actually cutting anything, but it's looking at individual layers of neurons. The cortex is made of many, many layers that are interconnected in this complex wiring diagram. And you can see the individual cells in shallow and deeper and deeper and deeper layers all simultaneously. And it's these neurons that in groupings of activity are controlling our behavior. So in the motor cortex, if we decide that we want to do a certain movement, then neurons in the motor cortex, these neurons, send a signal down to structures that are deeper in the brain in order to drive that activity. And as many of you know, that ability of that communication to actually induce the behavior depends very much on dopamine signaling, because in Parkinson's disease, where dopamine signaling is lost, the ability to initiate movement is hampered and eventually stops altogether, as you may remember from movies like Awakenings. So here we can again use light to read activity in the cortex of the mouse at individual cells. And then we can actually mimic the effects of dopamine by using light in order to control dopamine signaling. So let me just tell you how we use light to write signals to the brain. Dopamine is made by neurons that are deep in our brains in what's called the basal ganglia. And they come up into a central part of the brain called the striatum. And in the striatum, they converge. The dopamine signaling converges with signaling that comes from the neurons that you just saw active in the previous slide, those cortical neurons from the motor cortex. And there, the two signals interact, and then that controls the ability to induce movement. But it's complicated because the dopamine can act on 5 different receptors. And if you look at the brain, this is front to back, then you can see the different colored receptors, the different kinds of receptors, all to dopamine, are scattered quite broadly in the brain. And in the striatum, where you're controlling movement, you have almost every kind of these receptors. So to unravel what the dopamine is doing, it's complicated because it's acting on all these receptors and all these cell types and all these regions of the brain. The question is, can we get at that? And can we use light to do that? Here is the wiring diagram, right? The dopamine neurons that release dopamine in the striatum, the neurons from the cortex that come down and excite neurons in the striatum, what we call interneurons that receive information from the cortex, process it, and then the output neurons of the striatum that go and execute the movement activity. And if you look at them, you can see every one of these has some color dopamine receptor. So to unravel what's going on is really complicated. Each cell has its own receptors. There are 5 different receptor types. Is there any way that we can make coherence out of this? And the answer is that we use these light switches that we've developed. And what they consist of is basically something like a dopamine, but a version of dopamine that is targeted against one or another of these receptors. In this case, the orange receptor. It can only activate the orange receptor. It cannot do it in the dark. When you shine one color of light at it, then it changes its structure because of this little red elbow here. It doesn't look like an elbow, extension here. That enables the ligand to just become uncloaked, the blue thing, and bind like a dopamine would to the receptor to turn the receptor on. And if you shine another wavelength of light, another color of light at it, that folds this back into a different conformation, a different structure, which prevents that ligand, that dopamine-like ligand, from binding to the receptor and interrupts the activation of the receptor. So you can make the receptor active only when you shine one color of light at it, and when you shine another color of light at it, you stop it being active. So now imagine what we do. We target this thing to only a cell that we want because we put into that cell using a virus that produces common cold in us, but that is neutralized so it can't do anything other than deliver the gene. We put in this protein to which the photo switch, the light switch, attaches. That then goes to only one cell type. So when we look at all the cells in all the brain, we don't have to worry about them all. We have something that's only for the orange receptor and now is only for where we point the light and put in this switch, meaning only the cell types that we want. And it's something that you can turn on and off with speed. You heard from Max how quickly you can actually turn things on and off. You can also do this in microseconds or milliseconds, meaning at the speed that the nervous system normally operates. So when we do this, I'm just going to go back to this slide again of the mouse brain. Now we can pair when certain activity happens in certain neurons with activation of those dopamine receptors. And what we find is that whoever you pair that with, it ends up reinforcing that pattern. And that tells us something about how we learn to do motor behavior and how, if you ever make a prosthetic device that you want to control with the brain, because let's say you have a spinal cord injury, how you would design it in order to make certain neurons able to control that device, because the dopamine signal would reinforce a certain pattern that then would drive a robotic arm, for example. So we would copy the way the brain normally learns, execute this behavior, whether you're playing the piano or skiing, and use it instead to control a robotic arm. Okay, so light to read in this case, and with these chemical switches, light to write. And I'll stop there and take questions.
Thank you, Professor Isacoff.
So I had a quick question for you. The brain is a 3D structure. How are you using light to take pictures at just one depth?
Yeah, so that's a really excellent question. You know how you have a problem in pointing light in the atmosphere, and you solve it in your lab with adaptive optics and with laser pointing. So we can do the same thing. We can use these special devices called spatial light modulators to create patterns of light in 3 dimensions that will cover individual cells in 3 dimensions at various depths, despite the fact that light is scattered in the brain. And the way that we do that is by corrections using adaptive optics. So adaptive optics help for the reading of the activity in what otherwise would be a blurry kind of image, and for the specific, well-pointed writing of activity to specific cells.
Excellent. That's fascinating. If you have questions for Professor Isacoff, please enter them into the chat, and I think we'll come back to them towards the end of our evening discussion. That was excellent. Okay, so we'll now move on to our final speaker this evening, Brianna Peck. Brianna is an outstanding third-year undergraduate student working in my lab, and like me, she uses adaptive optics to improve the resolution of ground-based telescopes by correcting for atmospheric distortions. This adaptive optics works by measuring the motion of an image or wavefront and adjusting optics in real time to counteract that distortion. And Brianna is going to tell you about our plans for improving adaptive optics on the world's largest telescopes. Take it away, Brianna.
Hello, everyone. Thank you. My name is Brianna Peck, and as just mentioned, I am part of the Moving Universe lab, where I have been working under Professor Jessica Liu for a bit over a year now. My project in her lab specifically focuses on enhancing the image quality of the ground-based 10-meter telescopes at the Keck Observatory on the Mauna Kea Mountain in Hawaii with the light shaping technique called adaptive optics. Now, ground-based observatories need adaptive optics because of Earth's atmosphere. So specifically, when light is emitted from distant sources in our universe, it arrives to Earth as flat wavefronts. It helps to think of these wavefronts as flat sheets of paper. However, once the light enters our dense, turbulent atmosphere, these flat wavefronts become bent and distorted, and this effect is actually what causes stars to twinkle at night. If I go back to the paper analogy, it's as if our piece of paper, after entering the atmosphere, has become crumpled and balled up and then flattened again. So there's a lot of grooves and divots now in the originally flat wavefront. And when these bumpy wavefronts hit our detector, they appear as a blurry image. But with the technology of adaptive optics, we can recover the original flat wavefronts, and I can show you what I mean. So this image is an image of our galactic center without any adaptive optics. And it looks really blurry, and it's hard to pick out specific objects. But once we turn on adaptive optics, we can resolve out much more detail. Okay, so how is this done specifically? Well, an adaptive optics system starts by taking in the blurry light from the telescope, which bounces off of a mirror, which I'll get back to in just a second. This light travels through our system, where we split and we send most of it to our science camera to retrieve our resolved image. And then we send the other light into the first of 3 components of our adaptive optics system, the wavefront sensor, its job is to measure the so-called bumpiness of our wavefront. Then the second component is our control system, which calculates the correction needed to restore our flat wavefront. And then finally, we have the adaptive or also deformable mirror, which works in real time to correct the wavefronts we receive from the telescope. And this entire system works at 1,000 times per second, allowing us to keep up with the constant fluctuations in Earth's atmosphere. However, in order for our adaptive optic systems to work optimally, they need to be looking at really bright objects in our night sky. However, when the object we are looking at does not emit enough light for our adaptive optic systems, then we rely on stars around that object called natural guide stars. And especially dark regions of our sky, like the galactic center, where Professor Liu is hunting for black holes, It contains a lot of dust and gas, and these areas don't have enough bright stars around to help out the correction in the adaptive optics system. So we make our own guide stars with lasers, which we call laser guide stars. We make these fake stars with lasers that we shine really high up into our atmosphere, about 90 kilometers or so above Earth's surface. These lasers excite sodium atoms, which then shine their light back down onto our telescopes and create bright points of reference for our adaptive optics systems. This is similar to a CT scan of our atmosphere, where these lasers are helping us reconstruct a 3D structure of our atmosphere's turbulence in a process called tomography. And currently, each Keck telescope has one laser guide star and one deformable mirror. And this adaptive optics system works really well in infrared wavelengths, or wavelengths of light longer than what our eyes naturally see, which we instead perceive as heat. So what I just said in words, I can show in a plot. So on this plot, going from up to down represents the resolution of an image. So as you move down, that means the telescope can pick up finer details and therefore has better performance. Whereas if you move left to right, this is the size of your wavelength. So to the left is visible light and to the right is infrared. And when we turn on our adaptive optics system, we can see a drastic improvement in our image resolution in the infrared, which is the Keck plus AO for adaptive optics. But we do not yet have a good adaptive optics system for the visible light. And if we compare this to the resolution of the Hubble Space Telescope and the new James Webb Space Telescope, then we see that both of these space-based telescopes perform better in the visible light range, but Keck still has better resolution in the infrared because of the technology of adaptive optics. So in order to help Keck move further down on this plot and improve its resolution and be able to see finer details at shorter wavelengths, we want to build an adaptive optics system that can also correct in the visible range. So to do this, instead of just one laser guide star, we want to have multiple. And instead of just one deformable mirror, we also want multiple deformable mirrors. Multiple lasers will help us build a better 3D reconstruction of our atmosphere. And we also want our entire system to move faster than it is right now to keep up with the higher frequency of visible light than infrared light as it travels through our atmosphere. We call this new style of adaptive optics visible light multi-conjugate for multiple mirrors adaptive optics, or as we say, VIS-MCAO for short. And over the past year, my main project has been to run simulations for this future telescope to determine exactly what upgrades need to be made in order to achieve this performance. Out of the parameters, one of the parameters I've tested has been the number of laser guide stars. So this plot is similar to the one I showed earlier, where performance improves as you move down on the plot. So, and as you move left to right, that is an increase in number of laser guide stars, and the different colors represent different brightnesses of the laser. So from first rounds of simulations, we see we'll need at least 8 laser guide stars in order to achieve visible light adaptive optics. Now, if I go back to the plot I showed earlier with Hubble, Keck, and JWST, if we add in the new VIS-MCAO system with multiple mirrors and lasers, we see that it achieves better resolution than either of some of our best space-based telescopes, which I find really exciting. And to conclude, I just wanted to share some more simulations that I have been running. So at the beginning, I showed an image of with and without current adaptive optics. But this image right here is what a star would look like with, once again, the current adaptive optics system, but this time in the visible light. And if we were to build a new system then we would be able to achieve even finer resolution and really bring all of that blurriness into a really tight spot. And we call this type of spot a point spread function, and we really want to minimize its area. And overall, our goal is to improve the image quality of the Keck Observatory so we can see more of our beautiful universe. And I'm really excited and grateful to be part of this really incredible effort. And I'm also thankful to Professor Liu for her advice and guidance as I've been part of the Moving Universe Lab. So thank you.
Thanks, Brianna. And I promise I didn't pay her to say that. That was a fascinating talk. We do have a few minutes for questions for Brianna. If anyone would like to put those into the chat, go ahead. But I will kick us off. So Brianna, how powerful or bright are your lasers that you're using, those laser guide stars you were talking about? Or how do they compare to a laser pointer, for instance?
Yeah, so they're actually not super powerful. They're about as powerful as the average light bulb, so about 20 watts. And for the new system, we actually want to use slightly more powerful, but not crazy, about 60-watt laser guide stars.
Good. Well, we have another question then in the chat asking about laser interference with planes or rockets. Do we have to worry about our lasers shooting down planes or rockets or satellites?
Yeah, it can be a bit of a problem, but there's actually some techniques where some laser guide stars are made in the UV, specifically so they don't shine onto pilots or any satellites. But I'm not exactly sure how telescopes do it currently, but I know in the a few years ago, there used to be people outside actually spotting airplanes, and there'd be like a button that someone would press to let them know to turn off the laser inside. So.
Great. We have another question about how deformable mirrors work. How do you make those floppy mirrors that move 1,000 times a second?
Yeah, so it actually moves on such a small scale that even your eye has a really hard time seeing it. Like it moves about a micron, which is really, really small. And this is done with actuators. So we have like a flexible piece, and then we poke it with little pistons after we calculate the shape that we want it to go in, and it changes, as I said, about 1,000 or more times per second.
Excellent. And then I just have a quick question for you, Brianna. As an undergrad here at UC Berkeley, how did you get involved in research? And have you been able to go to any of the telescopes?
Yeah, so I got involved in research through a cold email. I was a freshman and was just really excited to be in the astronomy major. And I reached out to Professor Liu, and she was like, I have this adaptive optics project. And I was like, okay. And it's been really great ever since then. And yeah, this summer I was at Caltech, and I got to go see the Palomar 5-meter telescope in person, which was really amazing.
So Brianna,
Excellent.
The question is, when are you going to switch to looking in rather than looking out?
Oh.
Come to the brain.
Yep, adaptive optics is everywhere. That's right. Yeah. Excellent. Well, thank you so much, Brianna. And I think all of the presentations tonight have been fascinating. We have some time left for questions, so keep posting your questions in the chat. And we invite all of your questions for any of our speakers for tonight for some additional questions. So keep those coming. And I think we have a few rolling over from previous chats. So maybe I'll start with Udi. Can you tell us a little bit about what it's like to involve graduate and undergraduate students in your research? We have a lot of alums on with us tonight.
Listen, that's one of the beauties of Berkeley, that they're amazing undergraduates. Brianna's an example, and graduates like Max. I'd love to recruit them to our efforts, but they look like they're actually onto very good things. They don't need us. So, in my lab, we get students who come from a variety of different disciplines, from physics, biophysics, biology, psychology. Not enough yet from computational science, but that's going to happen more and more because we all need it. And there are a bunch of different graduate programs that all kind of interlace and that produce really fantastic students. So, I think everybody who's at Berkeley as a faculty member and has a very hard time ever leaving, it's because of the colleagues, but it's a lot because of the students.
That's great. Max, I think we had a lively discussion going on the chat about whether your laser switching techniques can be used to etch in tiny circuits into any 3D materials, and if those circuits could be made smaller than what we can make today. So I thought maybe you could tell us a little bit about that kind of application.
So you could certainly use a laser to etch tiny structures in your material, and then you basically just limit it by the spot size of your laser. And I mean, you can focus laser down like to incredibly small spot sizes if you want, like sub-micron. So this is like, with this kind of technology, we would definitely get to way smaller device sizes that we are currently at with transistors and these computer chips that are currently made. So this will, we can make incredibly small structures with this technique, yes.
And then a follow-on we had from the chat: if you were to make one of these fast-switching semiconductor circuits, would you have to adjust your laser input timing to be aligned with the timescale of any temperature changes? I don't know how stable the temperatures can be made.
I mean, that is like the biggest problem with this material at the moment, that it's kind of complicated to use it in technology, is because it really only works at really low temperature. So this kind of metallic state is stable below 50 Kelvin. And if you know, 0 is like the absolute 0, so that is a very, very low temperature. That is technologically very demanding. That's below liquid nitrogen temperature. So like this kind of experiment, you always need some kind of cryostat anyways to keep the temperature low, and then it's not that much of an issue in our experiment. But for if you want to use this in like an industrial scale, then people are definitely trying to stabilize the state at higher temperatures now. That's why we're doing like fundamental research and trying to understand better like why it is stable.
Excellent. And let's see, if I go on to the next question, we had a question about, does light travel easily through brain tissue, and will that work on live humans? I think you answered in the chat, but maybe you could say your answer out loud.
Sure thing.
Isacoff.
Yeah, the longer the wavelength, the easier it penetrates. And so if you have light that is blue, it gets scattered more than light that is red and infrared and so on. So longer wavelength penetrates better. The problem with longer wavelength is, can you get it to do something that you'd be interested in doing? And one thing that you can do with light is you can get molecules to absorb it even when they are designed for, let's say, something that's blue or green. Let's say something that's blue and would normally absorb light in the blue range, which is about 450 nanometers. It will absorb light also that is much weaker, that is red, infrared, at double the wavelength, because of a process that's called two-photon excitation, meaning two weak photons come and are absorbed exactly at the same time, and they add up to the equivalent of one blue photon. You can also do three-photon and so on, right? So there are tricks that you can play with light that penetrates much more deeply without scatter, and that still will excite things in the kind of wavelengths that we normally work with. But you can also design your antennas to work with much longer wavelength of light. And ultimately, what we're all going to need to do, especially when we want to go into humans and do things as non-invasively as possible, is to get forms of irradiation to do what I showed you today with visible light, but to do it with much, much longer wavelengths that can just go through the skull in a non-invasive way.
That's fascinating. I wish we could keep going with this fascinating discourse. I have lots more questions percolating along. Feel free to keep posting your questions in the chat and we'll try to get back to you. But we're almost out of time for the evening and I want to leave just a minute here to hand over the microphone back to Maria Hjelm to close this out. Maria, the floor is yours.
Thank you. And thanks to all of our speakers tonight. And thank you for sharing your research. These talks were fantastic. It's always really fun to find the common ground in very disparate fields. And I'm curious if many astronomers have gone over to neuroscience, for example. I could definitely see that happening. Anyway, to close out this evening's session, let me say a big thanks to our audience for joining us. We love to have you here. Your interest in the science we do is incredibly rewarding to our researchers and our students. And a reminder of 2 upcoming events. One, on October 18th, we will host our very first live version of In the Basic Science Lights the Way series during homecoming weekend. So if you're planning to come, please join us at the Chevron Auditorium in iHouse for a discussion titled "It's Getting Hot in Here: Insights on a Warming World," featuring a few of our Basic Science Lights the Way alums and 4 of our faculty members. Those details can be found on the UC Berkeley Homecoming website. And mark your calendars. Oh, it will also be streamed. So there'll be a way to view it online as well. And mark your calendars for Wednesday, October 30th, the day before Halloween, when we'll offer another virtual session called The Sound of Science: Connections Between Neuroscience, Physics, and Music. That one should be fantastic. And I encourage you to reach out if you'd like to learn more about anything that we covered this evening. As usual, a video of today's session will be available at the basicscience.berkeley.edu website about 2 weeks from now. Until we see you again, stay curious, fiat lux, and go Bears.